We have two great scientists talking to us, Doctors Natalie Krahn and Jim Van Deventer. It is my pleasure to first introduce Dr. Natalie Krahn, Assistant Professor in the Biochemistry and Molecular Biology Department at the University of Georgia. Dr. Krahn received her bachelor's in biochemistry and biophysics or physics at the University of Manitoba in 2013 and her PhD in 2017 in biochemistry, also from the University of Manitoba. She postdoced in Dieter Sohl's lab at Yale University from 2018 to 2023, after which she took an assistant professor position this fall at the University of Georgia. Her research, encompassing an impressive 29 publications, is currently focused on building tRNA libraries and using directed evolution to design and engineer translation systems for site-specific insertion of selenocysteine, nature's 21st amino acid, with the goal of ultimately harnessing the unique chemistries of selenocysteine to build new proteins with medical and industrial importance. Dr. Krahn, we're very thankful to have you with us today. Looking forward to your talk. Take it away.
Thank you for the introduction. So let me just this panel. There we go. Okay. So yes, thank you for the introduction. Today I'm going to talk about engineering post-translationally modified selenoproteins in E. coli, and I kind of give you a little sneak peek as to the technology that we use. And so this is a depiction of one of our engineered tRNAs. And we have it engineered so that it follows the somewhat natural pathway for synthesis of selenocysteine, where you first get cerylation and then that conversion to selenocysteine. And then from there, instead of using the natural elongation factor, we've harnessed, for selenocysteine, we've harnessed EFTU to install it into the ribosome. I'll kind of move forward with that and explain a little clearer throughout the talk, but this kind of just a nice visual representation of it. So as many of us know, the nature has a naturally recoded stop codons. So at least what I was taught in biochemistry is we had 64 codons recoding for 20 amino acids with 3 stop codons. But now we know that there are up to 22 amino acids, where the 21st amino acid recoded by UGA, and it's very fitting that I'm studying selenocysteine at University of Georgia, or UGA. And then we have the 22nd amino acid, which encodes for pyrrolysine. And so that's recoded by UAG. We see pyrrolysine in most archaea, some bacteria, and then selenocysteine we see in all 3 domains of life. And so really my interest is in selenocysteine, but you'll see in this talk how we've harnessed both of these systems and combine them together to make post-translationally modified selenoproteins. So selenocysteine, it's a small amino acid, but it's a very powerful amino acid. It looks similar to cysteine, but with this selenium atom. And if we think about, using this as a tool to expand our protein applications or studying proteins, it can be easily oxidized. And so one of the products of that is this diselenide bond. And this diselenide bond is very strong compared to disulfide bonds. You can also have a selenide sulfide bond, which is more stronger than a disulfide bond, but not as strong as a diselenide bond. And you can imagine that if you put this into a protein, that this can increase your protein stability, or you can use it to promote folding, especially in systems where there's multiple disulfide bonds. You can make sure that this one forms first, and then everything follows behind. Another synthetic application of this, or using selenocysteine as a tool, we can eliminate the selenium and give us this double bond. So it's a dehydroalanine. And so if we're able to site-specifically insert this, and we have an efficient way to produce dehydroalanine, now we can use this as a handle for post-translational modifications. And so that includes phosphorylation, methylation, acetylation, glycosylation. I know that there's been some work done on this, but because of the low efficiency of being able to use selenocysteine and insert it, I don't think this has been as studied or as developed for current use. But I think it has a lot of potential for that. And again, this goes to why hasn't a lot been done on selenocysteine. It really comes down to the translation and the unique translation system that it has. So canonically, we see that for all 21 amino acids, including pyrrolysine, you have a dedicated tRNA and a dedicated synthetase for that to put on the specific amino acid. And this gets brought to the ribosome using a natural elongation factor, matching codon anticodon, and then your amino acid is inserted into the ribosome. If we look at selenocysteine in bacteria, the pathway is a little bit more complicated. So we do have a dedicated tRNA, but now that tRNA first gets serylated by CRS, and then that serine is converted to selenocysteine via cell A. And this is because selenocysteine is very labile. It's not hanging around in nature in its reduced form. And so then selenocysteine-related tRNA is brought to the ribosome with a special elongation factor. And this in bacteria is called cell B. And this specifically recognizes the tRNA, the amino acid, and then this hairpin element called a cis element. And that's in the translated region of the mRNA. I told you that we see selenocysteine in all three domains of life, but all three domains of life, the translation pathway differs. And we look at eukaryotes, now the pathway has multiple steps. That conversion from serine to selenocysteine happens in 2 steps. And now there's extra binding proteins and partners in the elongation path of this. And so it adds complexity to the mix and really makes it hard for us to overexpress selenoproteins. It's hard for us to take a eukaryotic selenoprotein and make it in bacteria. And so we really needed a solution to this. And this is work that kind of stemmed from Dieter Sohl's lab. And I really harnessed it and continued on with it throughout my postdoc and into my own lab. And that's really focused on tRNA engineering and essentially creating a tRNA that is capable of putting selenocysteine on it, but using the natural elongation factor and not requiring all these specific factors that are required in selenocysteine insertion naturally. And so the scaffold we use for tRNA insertion requires this allo-tRNA, which we found in metagenomic data. And so we've engineered this to promote the selenocysteine insertion. And kind of just to basically look at the structure, we have this long variable arm, which we need for the first initial serylation. Conversion to selenocysteine requires this D arm to have multiple 6 base pairs and a 4 base loop. And then also to have this 12 base per acceptor domain for the EFTU binding. And so while I'm not going to go too much into the detail of it, we do have a system fairly well established in E. coli where we can use these allo-tRNAs and get insertion of selenocysteine via EFTU using the UAG codon. And one thing that we do have to be aware of is that we can get some serine put in, just given the fact that the elongation factor recognizes just an amino acid and not specifically selenocysteine. So this is great. We have a system for E. coli, and we have a tool to be able to install selenocysteine site specifically. But when we look at things naturally, in humans, we have 25 selenoproteins, and the role of them is essential to our survival. And so what we need to be aware of is that there's a correlation between selenoprotein deficiencies, mutations in these proteins, or selenium deficiencies. So we get selenium from our diet, which will then make your selenocysteine to make your selenoproteins. And so this is correlated with numerous diseases. These include neurodegenerative diseases, thyroid dysfunction. Keishan disease is actually the first disease associated with selenium deficiency in Keishan, China. Cancer, diabetes, cardiovascular disease, and inflammatory bowel disease. So there's a definite correlation between them, and this is why we kind of have like a black box, because we don't actually know what comes first. Does the disease come and then we notice these deficiencies, or do the deficiencies cause it? But in addition to that, as humans, we have a microbiome inside of us, and these microbes can also have selenoproteins. And if we have a selenium deficient diet, then those can affect the levels of selenium in the microbiome, and it also affects the makeup of the microbiome. And so, if there's a competition between selenium, now could potentially cause selenoprotein deficiencies in us, leading to other diseases. And there's been studies done which have connected selenoprotein microbiomes to some of these diseases that I've talked about, as well as some new things. So when we think about this, a question kind of comes up, well, we have 25 human selenoproteins. We have a system in bacteria to make selenoproteins. How can we go about and make eukaryotic selenoproteins in E. coli? Given the fact that we know that some of these eukaryotic selenoproteins have post-translational modifications. So this is really kind of what was the center of why we decided to combine our system with a pyrrolysine system to make post-translationally modified selenoproteins. So this project was done with under, I guess, my supervision as a postdoc to a grad student, Lance Nunez, a high school student, Cody Cummins, and an undergrad, Pearl Morosky. And so our goal was to take this allo-tRNA system I've talked about that introduces selenocysteine, and then take a pyrrolysine system that has been engineered to insert acetyl-lysine to make natural selenoproteins. And kind of the goal is to take an mRNA. And so, of course, we started with GFP because it's a nicer reporter. We can see the fluorescence of it, and install both the selenocysteine and acetyl-lysine at dedicated stop codons to make a post-translationally modified selenoprotein. So what we found out pretty early on, so if you're trying to install 2 amino acids, you need 2 stop codons. You need to make sure that the stop codon that you're choosing for your system is actually putting in that amino acid at that position. And we found out that when we used our selenocysteine system, a GFP reporter, when selenocysteine is supposed to go at UAG, we found that we got fluorescence both at UAG and at UGA, and they were pretty actually similar amounts. So we realized that the tRNA that we had was not overly specific for the codon that it should be recoding. We did the same thing looking at the acetyl-lysine system and noticed that the acetyl-lysine tRNA that we use, the pyrrolysine tRNA that's supposed to go in at UGA, the fluorescence was actually pretty low, and that tRNA preferred recoding of UAG. And so this is quite discouraging at first because we thought, okay, well, you know, our 2 systems are not working very well together. Kind of highlighted here is that acetyl-lysines with the anticodons to recode UGA is actually recoding UAG much better, and our selenocysteine tRNA is recoding both of them to the similar efficiency. So we simply said, okay, well, can we just swap the codons, and will that make a big difference? Can we shift that preference? And strikingly, that's what happened, and we were pretty happy with this result. If we put the anticodons to recode UGA for the selenocysteine, now we get nice read-through at UGA only and not UAG. And the same was true for the acetyl-lysine system. If we put that at UAG for recoding instead of UGA, then we got that nice fluorescence at that single codon. And so now we had a system that we felt was, we could combine the 2 together without getting the crosstalks. It can be nice orthogonal systems to one another to be able to make these natural selenoproteins. So we showed that it's using a reporter system that it worked, and that we were able to, you know, choose one versus the other. But the main thing was, okay, can we actually make the protein? Are we able to see the protein? Can we confirm by mass spec that they're in the right positions? And so because we had the construct already for GFP, we started with that. And we did use both tRNA systems just for comparison to see our optimized conditions where there really is no crosstalk and the tRNAs are recoding the correct codons. And then the original system where we got that mix. And we saw even with protein production that the yields we got for GFP production was significantly lower with the original tRNAs, and with our optimized conditions where we know or have a high confidence that the amino acid is going in at the right codon, that our protein yield was a lot higher. And so we were getting roughly 0.36 milligrams of protein per liter. But as we know, GFP, while it is a great reporter and it works well for all of our genetic code expansion techniques and optimizations, we really wanted to say, okay, well, can we actually use this to make natural selenoprotein? And so we kind of scanned the database to see what selenoproteins had post-translational modifications, which ones were kind of easier to work with, at least to start with. And so we chose to work with glutathione peroxidase. Glutathione peroxidase, I'll show you the crystal structure here, is a tetramer. Here we see the crystal structure shows it as a dimer, but you have a monomer of about 25 kilodaltons. Your selenocysteine is highlighted in burgundy or magenta here, and it's roughly around position 49. And then there are multiple acetylation sites. So I highlight 2 of them that we are interested in, but I think they're up to 4 or 5 of them. And so our idea was, can we, install the selenocysteine? Can we install the acetyl-lysine? And is there a preference for position? And are we able to kind of get pure protein this way? So we express a protein using our 2-plasmid system. And we had acetyl-lysine at position 114 and at 148. And what we noticed was that actually the distance between the selenocysteine, which is at 49, the further away we are, we seem to get a little bit more protein. So there might be some sort of balance there between trying to optimize. If we have a choice where we can put certain post-translational modifications, maybe we aim for something a little bit further away, might yield better results. So we were excited. We saw here our 25 kilodalton monomer, and then we saw these other bands, which, we thought, okay, this was a reducing gel, but it wasn't denaturing. So maybe some of the tetramers still stuck together. And so we assumed that there was some oligomerization happening here. But to verify and to confirm that this was the case, what we did was did a Western, and we used acetyl-lysine as to blot it, an anti-acetyl-lysine antibody. And we noticed that only this top band was luminescing, or we could see on the blot, and not this bottom band. And we were a little confused because we were pretty sure that this, it's the right size for the GPX one. It should be the monomeric version, but it wasn't showing up on the blot. And so we kind of went back and confirmed the efficiency of the acetyl-lysine system that we were using. And we found out that if we have the synthetase expressed and the tRNA expressed, but we don't add the amino acid in, we're still getting decent read-through and fluorescence. So something is getting inserted that is not acetyl-lysine, but that the system will pick up. So we have some nonsense suppression going on. And it's about 2/3 that of if acetyl-lysine is present. So from this, we can kind of maybe guess that only about 30% has acetyl-lysine, which, if you go back, kind of correlates with the intensity of these bands, where there's about 2/3 down here and 1/3 up here. And this was the same for both, regardless of the anticodon of the tRNA, whether it was recoding UAG or UGA or UAG. And we saw this also, confirm this to see what amino acid was going in using mass spec, and found out that indeed it was glutamine, which is a very common suppression that we get, nonsense suppression. So there was a lot of glutamine going in, but we did have acetyl-lysine. And I think what was very exciting for us, being interested in selenocysteine, is that we were able to get 100% selenocysteine put in, but we were now limited by the fact that the acetyl-lysine system was not overly strong, was only giving us about 30% of insertion. So with that, I'd like to kind of just wrap up and realize that through this project, we got some really exciting results. We were able to produce an acetylated seleno superfolder GFP, but maybe more interestingly, we were able to get glutathione peroxidase. We were able to show that we can put acetyl-lysine in at 2 different positions in addition to selenocysteine, and we had very good selenocysteine insertion. But what I think came out of this more was that we ended up with more questions than answers. Yes, we developed this technology, but at the same time, it brought about some very unique questions that I think we're interested in pursuing. And the biggest one that came was for this codon preference, and why one system requires UGA versus UAG. What is that preference? We like to think that synthetases don't have a preference for that, but maybe there is something inherent about these tRNAs that says, okay, well, this codon is not as specific for, or this anticodon is not as specific for the codon. And so we're kind of questioning this, and maybe this can give us some insight when, further development of tRNAs, what stop codons we should be using. And the other question that came up with regards to looking at these natural selenoproteins is, what is the role of acetyl-lysine in glutathione peroxidase oligomerization? So remember, all the acetyl-lysine was in the upper band, so those glutathione peroxidase enzymes are oligomerized. The monomers didn't have any acetyl-lysine, and so this provides us this opportunity to study these functional roles of the presence of these post-translational modifications. So with that, I think I'm pretty close to being on time. I like to thank the students that did this work. So this was all done at Yale, my postdoc, with collaborators or my previous colleagues back at Yale, Oscar, Christina, and John. And then I just like to thank my current lab. It's kind of been evolving. I've been here for a semester. Kind of started with the first rotation, third rotation of grad students, and then my lab here now in 2024. So I'm excited to keep working with this, and really interested in moving forward with selenocysteine, and I'd be happy to take any questions.
Great. Thank you so much, Natalie. It was a really great talk. It's not very often in the GCE world that we hear much about selenocysteine, so this is really fantastic. So are there any questions from the group? We got, I think, one here from Ryan.
Yeah, great talk, Natalie. Question on the very end there, you showed the oligomerization with regard to the specificity of acetyl-lysine versus not acetyl-lysine, right? And I didn't know if from your mass spec, you were able to differentiate what was going on there. Was it deacetylated, or was there a different amino acid there? I missed that part.
Yeah, so we saw glutamine there. So that was going in instead of acetyl-lysine.
Okay, so then that would be near cognate suppression of that system, and somehow it seems to only be localized to the monomeric form.
So yeah, so I think what happened is that, or this is my perspective on it, is that the acetylation is causing the oligomerization to happen, and somehow promoting oligomerization. And when it's not there in the glutamine version, then we don't see the oligomerization happening.
Yeah, no, that's really cool. I mean, that's kind of at some level, that's really exciting, right? Because now you have you have the PTM switch almost at some level elucidated to maybe how the biological function is working.
Absolutely. And I think now that we have the technology to site specifically insert this, then we can try to tune this and figure this out. And does it only need one acetylation site? Do we need to have multiple? Because as I mentioned, there are multiple acetylation sites on this enzyme. So, what is the role of all those, and how do they work with the function? And I think it's a surprising result that we got, that now it's, we want to further investigate and figure out what's going on.
Do you think that you would be able to get enough material to do some structural evaluation of what that acetyl-lysine is doing at interface sites, or protein interfaces?
I believe so. I think like I said, we're kind of limited with the acetyl-lysine incorporation. We're trying to work on seeing if we can get that fine-tuned to get more of that, because if we're making these protein and we lose two-thirds of it, that it's glutamine, then that kind of limits it. But I think the selenocysteine technology itself is good enough to give us enough protein for structural work.
Great. Nice talk.
Thank you.
Great. I'll ask a real quick question, just sort of follow up on Ryan's there. Did you, in your mass spec, maybe I missed it, did you see any deacetylation activity in E. coli with your protein? Were there any with just lysine present at that site?
No, we only saw acetyllysine or glutamine.
Okay. Good. That's good to know. I mean, we've had some collaborators that have seen a little bit of deacetylase activity in E. coli, and so that's great to know. My other quick question is, maybe you touched on this and I missed it. Did you ever try TAA for stop codon for encoding either of the other two amino acids and compare that with what you had?
No, so that's a great question, and no, we haven't tried that yet, but it's definitely something that I think we need to look at and start given the fact that there was this complete switch between the UAG, UGA, yes, like it makes sense that pyrrolysine, the natural codon is UAG, but we're also using we're not using a natural selenocysteine system. We're using an allotRNA system, which is not, the natural codon is not UGA. So that was kind of like surprising that, it preferred UGA versus the UAG. How UAA plays into that will be interesting to test, and maybe give us some further information as to, when we're designing these tRNAs or choosing which stop codon to put them in, what we should kind of be aware of in terms of which codon it prefers.
Yeah, great. Any other questions from the audience? I can ask one real quick one since we have a minute. The selenocysteine that you use, do you just add it to the media as the amino acid, or is there a biosynthetic pathway from some seleno-containing precursors, or how does that work for your system?
Yeah, so we add in sodium selenite, and so sodium selenite will then get converted by cell D to a selenophosphate, which then will be used to convert that serine to a selenocysteine.
And is there any reason to believe that could be rate limiting in the encoding strategy, or does it build up pretty high concentrations in the cell?
Yeah, so we add that in, and again, it's trying to, because it can be toxic to the cells, so we have to be careful when adding it. But we want to have enough to make sure that because if we don't have enough, we'll get too much serine in and not enough selenocysteine. And so we've been playing with adding it in when we express the tRNA, and then adding it again, dosing it again later, so that there's enough present to keep the selenocysteine there without overwhelming the cells. But there, and it also depends on protein levels. If you have a cell that produces a lot of protein, or it's a protein that's very highly expressed, we might need to work on putting more in, and cells that don't produce as much, we don't have to put as much. But there's not a set amount that we've used yet, but it's something we consider when we're adding it in.
Great, cool. And there was one question from the audience, but looks like you effectively answered that just now. So,
okay.
Great. Anything else from the audience before we move on to Dr. Van Deventer?